Good morning, students, and welcome back to the class. Today we will discuss about the chromosomal theory of inheritance. Okay, and in the previous class we discussed about uh, the law of independent assortment. Okay, with respect to the dihybrid cross. So today we will discuss about the chromosomal theory of inheritance. Okay, and before discussing this, that what is that chromosomal inheritance of theory of inheritance and all that, we will see regarding the Mendel's work and why his work was not accepted in the beginning and all that, and the reasons for that. Okay, and so how this chromosomal theory of inheritance was put forth, by whom it was proposed. So to prior to that, discuss this theory of inheritance, chromosomal inheritance, theory of this chromosomal theory of inheritance. We will discuss about the Mendel's work and why his work not accepted, and what are the reasons and all that, so that we will reach to this, that is a chromosomal theory of inheritance. Okay, so Mendel started work in the year 1856 and he worked up to 1863. That is Mendel worked from 1856 to 1863 for 7 years of long period on this pea plant. Okay, and he published his work in the year 1865. Okay, but his work was unrecognized till 1900. ನೋಡ್ರಿ, 1856ರಿಂದ 1863ರವರೆಗೆ 7 ವರ್ಷ ಏನಾಗ್ತದ, ಮೆಂಡಲ್ ಈ ವರ್ಕ್ he conducted experiments, and in the year 1865 he published his work, but his work was not recognized till the 1900. Okay, and what are the reasons for that? Okay, first thing is that communication was not easy during those days. During the period of Gregor John Mendel, the communication was not easy as today it is. Nowadays communication is so easy. At your fingertip you can communicate anything to any corner of the world within a fraction of second, but it was very difficult. The communication was not easy during those days, and that is why his work was not widely published. That is one reason. Second thing is that his concepts of these genes, that is factors which in the words of Mendel, okay, as stable, unblending, okay, and factors for various traits not accepted. Okay, you see, the second thing is that the Mendel's concept of genes, that is the factors, they are stable and they are unblending, that is they won't mix during gamete formation. Okay, they independently they separate, they segregate independently. Okay, and there are factors of which are responsible for various traits and character, that was also not accepted. Okay, and Gregor John Mendel used the mathematic logic and statistical analysis to analyze these results. Okay, so another reason is that Gregor John Mendel was the first to use this mathematical logic and statistical analysis, okay, to analyze his experimental results. And this use of this mathematical logic and statistical analysis was very new during those period, and that is very new to the biologist during those period, and that is why it was not accepted. So that is another reason. And finally, Gregor Mendel's work suggested the genes or factors were the discrete units of the inheritance. Finally, the Mendel's work or experiments, it suggested that this genes or factors were the discrete units of inheritance. Gregor John Mendel, he proposed that the genes or the factors are the discrete units. They are the units of inheritance of characters from one generation to another generation. Okay, but he did not give any physical evidence for the existence of these factors. But he proposed, he suggested that the genes or factors are the discrete units of inheritance, but he did not give any physical evidence for the existence of these factors. Okay, and that is why his results of experiments were not accepted. So this is another reason. Okay, fine. Next, what happened in 1900? In the year 1900, Mendel's work was rediscovered independently by 3 scientists. You see, in the year 1900, okay, the Mendel's work was rediscovered independently by 3 scientists, and the scientists were the Hugo de Vries and Correns and von Schermat. Okay, so in the year 1900, the Mendel's work was independently discovered by 3 scientists, that is Hugo de Vries and Correns and von Schermat. These 3 scientists, they independently rediscovered the work of Mendel. Okay, and during this period, that is during 1900, what happened? There was advancement in the microscopy. That is microscopes were developed. That means microscope antandre Sukshma Darshaka Yantra. The equipment which can be used to see the small objects or the objects which are not visible to the naked eyes. Okay, so in the year during this period, that is during 1900, what happened? There was development in the microscopy, and microscope was invented, and invention of this microscope that led to the discovery of structure in the nucleus. Okay, so due to this microscope, that the scientists found a structure that in the nucleus. They could able to observe the cell and nucleus, and they could discover some structure in the nucleus. Okay, and that appeared to be double and divide just before the cell division. You see, due to the advancement in the microscopy and due to the development of microscope, the scientists could able to study the cell in a very minute way, in a detailed way, and they could able to study the nucleus. Okay, and they discovered a structure in the nucleus of the cell, and that structure appeared to double. That means double ಅಂದ್ರೆ ಹೆಚ್ಚು ದುಪ್ಪಟ್ಟಾಗ್ತಿತ್ತು, and it divide just before the cell division. The scientist who discovered a structure in the nucleus that was used to be double and used to this divide before the cell division. ಸೆಲ್ ಡಿವಿಷನ್ ಆಗೋಕ್ಕಿಂತ ಮುಂಚೆ ಆ ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ. ಈ ನ್ಯೂಕ್ಲಿಯಸ್ ಅಲ್ಲಿ ಒಂದು ಸ್ಟ್ರಕ್ಚರ್ ಏನಾಗ್ತಾ ಇತ್ತು, ಡಬಲ್ ಆಗ್ತಾ ಇತ್ತು ಮತ್ತು ಅದು ಡಿವೈಡ್ ಆಗ್ತಾ ಇತ್ತು. ಅಂತ ಒಂದು ಸ್ಟ್ರಕ್ಚರ್ ಏನಾಗ್ತದ ವಿಜ್ಞಾನಿಗಳು ಕಂಡುಹಿಡಿತಾರ. ಅಂಡ್ these structures are called as chromosomes, you see. So then they come to know that these structures in the nucleus were chromosomes, okay. And further in the year 1902, this Walter Sutton and this of Theodore Boveri, this in the year 1902, two scientists, that is Walter Sutton and Theodore Boveri, they noted that the behavior of chromosomes was parallel to the behavior of factors or genes. You see, in the year 1902, two scientists, that is one scientist is this Walter Sutton and another one Theodore Boveri, they observed or they find out that the behavior of the chromosomes chromosome was parallel to the behavior of the genes or traits which the Mendel found. Okay, the parallel means similar. That means this Walter Sutton and this Theodore Boveri scientist, they discovered or they noted the behavior of chromosomes was parallel or was similar to the behavior of the genes or factors found out by the Mendel. Okay, so then they gave the chromosomal theory of inheritance. Okay, so the factors, okay, what where the discrete units of inheritance as proposed by Mendel were the chromosomes. Okay, and that is why this Walter Sutton and Theodore Boveri, they put forth the chromosomal theory of inheritance. This means the inheritance that is a transfer of these characters from one generation to another generation is because of these chromosomes. Okay, so that is what the chromosomal theory of inheritance says, that and this chromosomal theory of inheritance was put forth by this Walter Sutton and that of Theodore Boveri in the year 1902. Okay, and these scientists, they used chromosomal movements during to explain the Mendel's law. Okay, and all of you have already studied during cell division how the movement of these chromosomes takes place, how they get separate from each other. Okay, so based on that, these scientists they proposed this chromosomal theory of inheritance. That means what this Mendel said, these factors or genes are responsible for inheritance. So that is why he put forth the Mendel's theory of inheritance. So those factors or genes are nothing but the chromosomes. That is why they put forth the theory of chromosomal theory of inheritance. Okay, and this was explained, okay, based on the chromosomal movement during the cell division. Okay, so tomorrow we will see the cell division. Okay, that is mitosis and meiosis, how this chromosomes movement is seen during the cell division. Okay, with this we conclude here and continue tomorrow. Thank you.